Neiman et al., 2010. If you guys are interested in seeing more of these papers, I got them on a laptop in the back there. Anyway, this was really nice because the participants allowed the study to take muscle and blood tissue for biopsy, which is really important because working with humans is hard. You're not allowed to just do whatever you want, right? But they were really great. They probably paid them a little to get their tissue, but whatever. We're in a different age now. Okay, so what they did is they did see increases in several of the mitochondrial biogenesis genes. They saw sirtuin activation again, a couple of other biosynthetic genes for the mitochondria. What they did in this one is increase the distance at 60% VO max trial. Okay, that's where you hook a dude up to a bike, you put this little thing in his mouth, and you give him 60% of his total oxygen intake. Okay, what you give him is the VO. That's what we mean by 60. VO max is the velocity of air coming from the lungs. Okay, so they monitored the amount of air that was coming out at 60%, and they kept him there. They said you're going to work at 60% efficiency, and we're going to run you for 20 minutes, and we're going to see how much distance you cover. Okay, and one thing that happened in the quercetin group was an increase in the distance available at that oxygen intake. Okay, so they showed better endurance at a certain oxygen intake level, which means the mitochondria within your cells were working better because they absorbed more of the available oxygen, and you were able to go farther, which means they converted it into energy more efficiently, right? So now we got some human indication. The Merck, this is 2010, there have been some other studies before this on horses, kind of an interesting one on horses, race horses. Anyway, at this point, if you look up on quercetin on the Webernet, and you look to buy it, you will be bombarded with hundreds of people trying to sell you pure quercetin. And it's meant for athletes. If you consume it after or before a workout, it gets in your bloodstream better, you have more efficiency when working out. Some people use it as a recharge to get their mitochondria, get themselves back to where they were, like after a workout. Some people eat it before to get the effect of mitochondrial biogenesis while they're actually exercising. But you will find it out there, and your fruit has a considerable amount of it in different forms. Not in the pure form that they're showing here, but in glucoside form, which is actually more available to you anyway. Plus, you're going to be able to convince people that eating a fruit is probably better than eating a supplement any day of the week. Okay, one final note on the whole antioxidant in a tube versus in the body thing. In 2010, you guys probably noticed that all the studies I cited were prior to 2010. In 2010, before 2010, the USDA had a huge database about ORAC test, and this is a general antioxidant test. It's just like I was telling you. You put the radical in with the extract, you see how long it takes to dissipate. Okay, it's called an ORAC, oxygen radical antioxidant capacity. Do it in a little machine. I can explain how you do it, but it's not that big. It was the be-all, end-all for years. What was your ORAC value? That's all people used to ask you. What's your ORAC value? Hundreds of foods, everything, fruit, cereals. Quaker oatmeal bars, you name it, they had it. Banana juice, everything. Thousands of food groups. Anyway, mysteriously in 2010, the database was completely removed. And when you went to the page which linked to the database, you got this. There is no evidence that the beneficial effect of polyphenol-rich foods can be attributed to the antioxidant properties of those foods. The data for antioxidant capacity of foods generated in vitro, test tube methods, cannot be extrapolated to in vitro. Basically what I just said to you. Effects and the clinical trials to test the benefits of dietary antioxidants have produced mixed results. They still have, by the way. It's not clear, cut and dried. We know how the antioxidant molecule in foods have a wide range of functions, many of which are completely unrelated to the ability to absorb free radicals. That's basically what I just told you. Right? So the USDA, the biggest, the baddest, have decided since 2010 that this whole antioxidant capacity thing is not worth putting the pixels up for it. I don't have to tell you guys how cheap pixels are. Super cheap. So they took it all down, said we don't even care. Don't even bother, don't reference it, don't talk about it. So anyway, there's the stance of the educated community on antioxidant tests as of late. Here's some of the general health benefits. I didn't go through all the compounds and all the benefits because we don't have time. Anyway, the other thing I wanted to highlight here is that while all these things do have a human effect, they have a role within the plant too. And the role within the plant generally gets completely disregarded for whatever they do in humans. I think it's the nature of the medical community. But anyway, some of these are quite interesting, especially the fact that the plant that you're consuming the fruit from still has these compounds in there, and they still do things while they're out in the field. We just, at this point, most people aren't interested in what the plant uses them for. They're more interested in what happens when you eat them. Okay? I happen to be more interested in this, but I might be the only one. One interesting part about the quercetins is they actually do a similar thing in both plants and animals, which should go back to show you how fundamental they are to mitochondrial biogenesis. Okay, So here's the take-home message, it's pretty quick, eh? Two important factors: the type of compound being consumed, the ability of the compounds for absorption within the body, and in vitro assays are junk. Unless you can measure in total content of something, then they're reflective of at least something, right? That's the amount of this class that's in there. When they're simple antioxidant challenge tests, they're not much. Okay, cool. So I just went through the polyphenylpropanoids, but you guys have a real cool fruit, see? And you guys actually have another set of metabolites in the fruit that are also health beneficial. Okay, they're called terpenoid compounds. They're from a completely different pathway that does a completely different set of things, and they're very interesting. Our lab, me, Bob, and our lab has recently discovered these two terpenoid compounds in the fruit of Larissa cerulean. I've been trying to release the paper for a while, but it seems like every time I get to it, somebody wants to put something new in there, and it keeps getting bigger and bigger. Anyway, we'll see what happens with it. In plants, these things act as herbivore deterrents, which is really great for the health beneficial people because nobody's going to put stupid labels on them and try and figure out what they are before they actually investigate it, which is great. Okay, so they didn't say, oh, these are more antioxidants everywhere, they're great. Okay, these don't act as antioxidants, they don't do it in the cell, and they ain't going to do it in your body either. Okay, so these are the two guys here. When GLC is just a glucose molecule. They have a couple of different activities when consumed by humans. We're going to talk about loganin because secologanin is not so well understood its pharmacological activity. Okay, so we'll stick to loganin. But it has actually a fair amount of research. So loganin, this little iridoid terpenoid, and memory impairment. They took some mice, they're always messing with the mice, and they induced a learning or memory impairment by scopolamine. Okay, this causes a euphoria in mice, gets them messed up in their heads. Okay, and then what they did is they actually fed one of the groups of mice, after they gave it scopolamine, they gave one of the groups of mice loganin, and they gave the other one nothing. Okay, and then they ran them through passive avoidance and water maze tests, which are just an ability of the mouse to figure out where it's going. They showed that the mice that had been fed loganin were actually able to pass the water maze test faster and were better at avoiding. Passive avoidance is where you put a food with like an electrode on it and it shocks the mouse and you got to figure out how long it takes the mouse to figure out that he shouldn't eat it. Yeah. Anyway, they scored better in these passive avoidance and water maze tests. The paper outlined that there may be therapeutic value from loganin in Alzheimer's disease. Okay? And this is backed up pretty quickly by a further study by Young et al. And gave evidence that loganin inhibits B secretase. B secretase is the enzyme in your brain that deposits amyloids, which is the cause of Alzheimer's disease. So they showed in vitro that this particular compound will actually inhibit this enzyme pretty specifically. And so one of the things they're looking for is possibly using it as an alleviation tool in Alzheimer's patients. This is the active concentration that you need to inhibit the enzyme. That's 2.9 times 10 to the negative 5 moles. I should have done it up for you guys. But that's very little. Okay, that's about keep 10 to the 5, you're around like 9 millionths of a mole or something like that. So it's very low concentration. Okay, so that's not the only thing they've shown though. They've also shown some effect of it in liver impairment in diabetics. Okay, what happens in diabetics is that they have liver impairments due to the insulin blockage and whatnot, and they have there's a certain microvascular complication that comes up. Anyway, it's called diabetic neuropathy. So they've showed that feeding loganin to patients in cell culture may have actually shown that they can reduce this microvascular complication. And another guy showed a year earlier that it actually helps fight oxidation in the liver in type 2 diabetics with the same diabetic neuropathy. Reduced inflammation, atopsis, a whole bunch of stuff. They did that one in mice as opposed to a cell culture, so it's a little bit better. Right on. So those are the compounds I wanted to talk about. Again, not horticulture. Keep in mind I'm a horticulturalist. I'm not a medical professional or whatever. We just have to, if you're going to dabble in this type of science, then you have to know what they do in the body, because you can get railroaded pretty bad if you're not really sure, like, what does this compound actually do? Is it an antioxidant, blah, blah, blah. So I'm not saying that I come up with any of this research, and I certainly don't do it. I don't like working with animals at all. So I don't like to do this kind of stuff, but you do have to have an understanding of how it works, or they get you. Anyway, for you it's important because you need to know what you're actually marketing, right?